And welcome. Hi, folks. So we're doing a little molecular genetics discussion, and I am busy working on our first sort of virtual PCR experiment, looking at bacteria ribosomal RNA sequences, and we'll see how that goes. And we still need to talk a little bit about PCR type experiments and setting up PCR, but we're going to save that probably for later this week. And so we were discussing last time some of the ideas about gene cloning technologies and the notion of doing a hybridization and the ability to use, to take advantage of base pair mismatch to clone genes from closely related organisms, closely related species. And it works. So you can, pretty much within all vertebrates, you can clone anything that way. Going from invertebrates like flies to humans, that requires some low stringency screening a little bit more carefully. Then there are some differences when you're trying to go between, really diverse species. I mean, years ago we were doing some work with the hermit crab and using gene sequences from both shrimp and fly, and we got some of the conserved pieces of the genomic DNA, but we didn't get everything. Anyway, so what we're going to do today is talk more about how genes encode RNAs. RNAs then are going to be translated into proteins, and some of the ideas behind the basics of genetics and how we can compare that to molecular genetics. So first off, let's start with some general terms as well to use in the cloning biz. We have two distinct types of sequence information for a gene. The first is pretty easily understood, and that would be the genomic DNA, the genomic sequences. Genomic DNA means a portion of the chromosome. And from a technical perspective, these days I would clone a chunk of genomic DNA by designing some PCR primers from a closely related species and isolating DNA and amplifying it. And that works fairly well. A classic way of identifying genomic DNA is, and this is a term that we use somewhat a lot, is building what's called a genomic library. And there are a number of interesting little parts to this. A genomic library is essentially a collection of clones that represent a genome. And trying to get a complete genome's worth of individual clones becomes a bit of a statistical game, but you can do this. There are a couple of standard ways of doing it. This is going to be just sort of a generalized way of doing it. There are a lot of tweaks to this, but this will get us kind of going on the right track. Just like we did for a genomic DNA blot, we're going to isolate genomic DNA. Now, the next step is to break it into small pieces, and a standard way of doing this is to take a restriction enzyme, rather than carry the restriction digest to completion, like let it go 37 degrees overnight, we might let it go 37 degrees for 10 or 15 minutes. And actually what you would do is set up a restriction digest, start the restriction digest, take aliquots out, and put them into a stop buffer, which is like something that has a little bit of SDS in it, and heat it up. And so you kill the enzyme. And you do that every 15 minutes. And so you'd have stuff that was barely digested and then stuff that was getting more completely digested. And you end up with a pretty distinct size range of DNA fragments. Now, if you're using restriction enzyme, because the other way of doing this is actually just to shatter the DNA either by passing it back and forth in a syringe needle or putting it in a sonicator, an ultrasonic tip, and whizzing it up. And then you're just shearing the DNA. In this case, though, you're cutting it, and what we can expect is that we're going to have the restriction digest ends. So each fragment is going to have the appropriate restriction digest end. And if you recall back when we were talking about restriction digest last time, the restriction digest EcoR1 has a restriction site GAATTC. It cuts asymmetrically, so you get what is known as a complementary end. Just to remind us of that, GAATTC is a palindrome, and it cuts like that. So it cuts at that GA, and like that. So you have the DNA, when it opens up, you've got this AT or TA overhang, and that's referred to in the biz as a sticky end. There are restriction enzymes that'll cleave and produce a blunt end. Cloning with a sticky end is way easier because you got a little bit of nucleotide base pairing to kind of hold the DNA fragment in place while you add DNA ligase. So we have restriction digest ends. The next step is to find something that in the biz we refer to as the cloning vector. The cloning vector is a piece of DNA that allows us to amplify and reproduce this DNA in generally bacteria. There are things that are called yeast artificial chromosomes, and you can carry out the amplification and replication in yeast, but that's not really used all that much. Primarily, the vectors are bacterial-based, and there are a couple of different bacteria vectors. A real easy one is something called, as we know, a plasmid, a circular DNA, which hopefully you recall from our work in molecular genetics or in molecular cell, or just live on the street. A plasmid is a circular piece of DNA. It's got the origin of replication on it for bacterial DNA replication. It also typically has some kind of antibiotic resistance gene, a marker like ampicillin resistance. And depending on the plasmid can replicate within the bacteria cell every time the bacteria replicates, or it can actually be a non-constrained plasmid, and it can replicate several hundred times within a bacteria. I like those a lot because you get lots of DNA. So you have the plasmid. Now, there's a lot of different kinds of games with this. There are also DNA vectors that are based on bacteriophage, and we'll be talking about those kind of things later. The main point is how much DNA can you fit into a vector? And plasmids probably have as their upper limit about 10,000 bases, about 10 kb. Beyond that, they're not very stable. So really big plasmids are hard to manipulate and mess around with. And one of the games when you're making a genomic library is you want to have reasonably large chunks of DNA in your vector, ideally about 40 or 50,000 bases. Now, depending on the genome size of your organism, that's how many individual recombinant clones you have to start working with to get a representative fraction of the genome. The usual, you can do statistics on it, but generally, if you calculate up the amount of DNA that you have in each vector, so the average insert size, and you know the genome size of the organism, if you've got approximately 3 times the genome size, then if you played out 3 times as much DNA as you have genome, you have a pretty good chance of being able to find your piece of your genomic DNA. So genomic cloning is really just become sort of a numbers game, and the organism you work with, depending on its genome size, can be really a lot of fun to play with, or it can be a real hassle to play with. Flies are a really great model system because they've got a fairly small genome size compared to humans or mice, compared to vertebrates. So you might have to screen through, say, 100,000 clones to have a representative 3x coverage of a genome in flies, but you might have to screen through maybe 1 million clones to have a representative genome size in humans. So that's one of the reasons why model systems are really very nice, and humans ain't a very good model system. So we have a gene of interest. We want to know about where it is in the chromosome, what its genomic architecture might be. We screen a genomic DNA library, and just to very quickly go through this, again, using PCR is sort of the modern way, and of course, if you're working on humans or flies, you don't do this anymore. You go to the computer because the genome is sequenced, and then you can get the entire piece of DNA that way. And we'll be looking at some of the tools, the computational tools, for looking at and annotating genomes. The vitamin C stuff I've been working on is a really good example, and we're going to be making some PCR primers for that here next week or so. But for our purposes, sort of the way we begin to do this, we would carry out a genomic library screen. So again, we make a radioactive P32 labeled probe, and say you found a gene in flies and you want to find its homolog in mice. Sure. We're going to have to screen a lot, so you make a mouse genomic library, or better yet, you buy one or have a friend give you one, and you make a P32 labeled probe. And then the way you carry this out is you plate out the recombinant bacteria to where they're not quite touching. You will need to see individual colonies, but it's pretty thick. And much the same way we did a DNA blot, you take a sheet of nitrocellulose, and they sell them in circular discs just for this purpose. You plop it on the plate, you peel it up, and what I like to do is replica plating. That way, you have a better sense of, is this a signal, is this a dot, or is this a piece of dust? So then you do a second one, and now you keep the plate as the original. When you put your discs on there, you make sure you're making little alignment marks with a pen, with like a needle, so you can line things back up. And then you take those nitrocellulose membranes and you put them into a high salt buffer with a fair amount of detergent, SDS, and then into sodium hydroxide. And what that's going to do is lyse the bacteria, denature the DNA, and the DNA is actually fairly well stuck onto the filter. And what I used to do is take a little chem wipe on my paper towel and actually polish the membrane to get rid of all the crud and bacterial debris. And you wash it a few times in a salt solution and then dry them. And then you can either use UV light or bake the filters to fix the DNA really well to the filter. And you've got a set of filters now that represent the DNA from these individual colonies. All right, you then carry out your hybridization, same way we did with the DNA gel blot, where you have your high buffer, you put it in overnight, you have conditions that are going to be below the melting point, and then you carry out the washes. So this thing keeps on kind of stopping on me. I have to figure out what's going on with that. But regardless, I think my voice keeps on going. Hope. But it's a little bit disconcerting when I'm looking at myself and I freeze. Like, I'm stuck in time. Oh, shit. But it's all right. So if you're doing, like if you've got a fly gene and you need to get a genomic library or a genomic clone, you can do a high stringency screen. If you're taking a fly gene and you're trying to identify something in a mice, you're going to do a real low stringency screen. So you're going to do washes that are 15, 20 degrees below the TM. And that way you'll favor the base pair mismatch. Now you're going to have more background and stuff, but that's all right. We'll deal with that. So then what you'll end up with is putting this on x-ray film, having a circle, a filter, and you'll see a spot. Now, hopefully, on the duplicate, you'll see a spot in the same spot. And when you do your little happy dance because you probably have got your genomic clone. You then take a toothpick or some kind of the back end of a pipette, and you jab and grab hold of that little piece of the original plate. So you line everything up, grab that plate, plop it into some media, get the bacteria, and then you carry out rescreens. And it's the same thing. You plate out the bacteria, you use the probe, and you identify, and the next time you should have, maybe not everybody is lighting up, but you should have a lot more dots. And you do replicas again, and then at that point, hopefully you don't have plates that are so thick that you can actually get in there with a toothpick and grab just that one guy and plate that out, and you now have a purified recombinant bacteria carrying your chunk of genomic DNA. Yay, you. Now, once you've done that, you can begin sequencing it, analyzing the exon-intron boundaries, all kinds of stuff. There's all sorts of things you can do. And so with a genomic clone, this is the information that we can get from it. So a genomic clone, we expect to see this kind of information. Now, it's not going to be all necessarily on a single genomic clone. It may take a couple of overlapping clones, or you'll have your gene embedded in 50,000 nucleotides and the other 45,000 nucleotides are irrelevant, but you got this. You're going to have +1, which is what we refer to as the first nucleotide of the RNA transcript. That is the beginning of transcription. And then you will have the upstream control sequences. And this is stuff that we call, of course, the promoter, and various kinds of regulatory sequences. Remember we talked about these in Molec and Cell. Enhancers. These are sites where transcription factors bind, and they can activate or block transcription. These are the control elements in the eukaryotic system. Now, because we're working with eukaryotes, we would expect that there's going to be regions of this portion of the transcribed portion of the gene that will actually be spliced out. That we're going to have exon-intron splicing going on. And so we refer to this as the structural gene. And it produces an RNA that is unprocessed, and it is essentially the pre-messenger RNA. So we're then going to have processing. And this is always the point, it's a little awkward in class, where I say, okay, somebody tell me what are the steps in RNA processing in a eukaryote. And I'm thinking to myself, people, we just went over this back in April, or March, I don't know, before we left. I think we did. Come on now. And it's like quiet, crickets, and thinking, oh, great. But I just spilled coffee. You can look it up very quickly, and you'll remember, processing. We've got exon intron splicing. We've got the addition of the 5' 7-methylguanosine cap and the addition of the 3' poly A tail. And you'll all say, oh yeah, I remember that now. Good. And so that is what happens when the structural gene is transcribed. Okay, so the reason a genomic clone is useful is that it can tell us some things about exon-intron splicing. It can give us the information about the potential for alternative splicing. It can give us the control sequences, because think about this, is a really cool experiment where we've got a gene that's expressed in a very precise spot in the heart. We can take the upstream control sequence and, using recombinant DNA, put something like green fluorescent protein downstream and now have green fluorescent protein being expressed in that spot. And it gives us tools to now begin looking at the mechanisms of gene regulation. So that's one thing you can do. Or you could take this gene and using a different promoter system, you could express it in different parts of the body. Or we could take a normal gene and put it into somebody that is carrying a mutated genomic gene and rescue them from, say, cystic fibrosis. So genomic clones are a useful tool. Now, another tool that is not quite as easy to conceptualize, but it's really important, and it's introducing the vocabulary we use in molecular genetics, is something known as a cDNA. So we've got the genomic clone and all of its information. Now, what is at least as important is identifying the messenger RNA that this gene produces. Now, remember, the messenger RNA is going to be processed. And here's one of the real tricks, is that with genomic DNA, pretty much every cell has the identical genome. Now, there are some differences, but immune cells actually undergo recombination of the genes for producing the immune proteins for the antibodies, and red blood cells in humans don't have DNA yet. Whatever, let's not get bogged down in those details. For the most part, every cell in your body or the organism you're working with is going to have identical genomic DNA. But genes, on the other hand, are not expressed in every cell. Now, if in the early days, what people were doing was cloning genes that are expressed at real high levels, like in muscle cells. So you scoop out a little bit of muscle and you clone actin because it's going to be like 20 or 30% of the total RNA is going to be actin. But if you really want to get interesting genes, they are not going to be expressed as highly. And so here's the trick. When you're trying to work with messenger RNA, one of the things you might do is isolate RNA from different tissues or from different times in an organism's development and do an RNA gel blot, a so-called northern blot, but I don't use those terms. And RNA is a tricky beast. RNA is a lot more sensitive to degradation, to RNases. It can be broken down and digested much more easily by accident. And so we have to be much more careful. Whereas genomic DNA is very stable, RNA is not very stable at all. Just as an aside, it's one of the issues when you're dealing with COVID is that it's an RNA virus, and to be able to detect it, you have to maintain the RNA. And the same thing with some of these RNA-based vaccines. The RNA can get into cells, it gets translated, and then the protein gets squirted back out. But the RNA itself has to be kept very cold, and so in terms of delivery and maintenance, it's not all that easy. But RNA is delicate. RNA to isolate it is not all that easy. We don't typically do RNA isolations around here just because it's a massive pain. Additionally, the gene of interest isn't going to necessarily be expressed in every single cell type or in whatever cell type it's supposed to be expressed in all the time. So we can make a library of messenger RNAs. Now, the critical point is you can't clone RNA, single-strand RNA. We have to turn this into DNA. And we're going to make something that is known as a complementary, and I misspell complementary, but I don't care, complementary DNA, known in the biz as a cDNA. So a cDNA is the messenger RNA in DNA form. How do we make that? Glad you asked. Again, this is one of the choke points in the COVID-19 testing procedure, is you have to take the RNA and you got to turn it into DNA. And the only way to do that is to use an enzyme that is called RNA dependent DNA polymerase. And let's unpack this term for a second. RNA-dependent DNA polymerase. We're going to see that's a really weird beast, and we really don't have that enzyme. Now, a DNA-dependent DNA polymerase, that's what you use to replicate your DNA. DNA polymerase, we all got that. A DNA-dependent RNA polymerase, that's what we call RNA polymerase. That's what you use to take DNA and turn it into RNA, to transcribe it into RNA. But an RNA-dependent DNA polymerase, that's a weird beast. That's going to take an RNA molecule and replicate it to produce a DNA molecule, a DNA copy. And I will just tell you that in our systems, we really don't have anything that does that. This is pretty much the realm of viruses, and specifically retroviruses, RNA-based viruses. RNA viruses, they carry out their lives as a genome that is made up of RNA, double or single-stranded, but it's still RNA. In order to become infectious and actually infect a cell, the RNA gets into the cell and it is going to be turned into DNA using RNA-dependent DNA polymerase. The enzyme is colloquially known as reverse transcriptase. And of course, in the molecular biology biz, we abbreviate this as RT. Now, the problem with this abbreviation is RT stands for room temperature, and even more problematic, RT these days stands for real time. And so when people are talking about doing real-time PCR, which is one of the tools that's used to detect COVID, and we'll be getting into this as we're talking about PCR technology. They do an RT-PCR, but it's actually RT-PCR. It's reverse transcription first to generate a chunk of DNA, and then they carry out RT-PCR. Not that anybody cares. All right, so reverse transcriptase. What we now do is isolate RNA, and using reverse transcriptase, turn that into DNA. And one of the things, whenever we're trying to make DNA, we always have to have a little piece of existing 3' hydroxyl to add on to. And this is known as a primer. And it's one of these things, every time I think about this, I go, why is that? I need to look it up. When we're making RNA, we don't need a primer. But DNA, you do. So you need to have a primer. And what we're going to use is a primer that we know will be part of a messenger RNA. So let's just draw schematically a messenger RNA. Here's the 5' cap. We've got the exons present. The introns have been spliced out, the intervening sequences. And now we've got the poly A tail. And the poly A tail is a great place to use as a primer site. We can use oligodeoxythymidine, a set of T's, T, 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 T. And remember, it's going to be running in the opposite direction. This is the 5' end. This is the 3' hydroxyls here. This is anti-parallel 5' and 3'. And so we always polymerize in the direction of 5' to 3'. So we start adding DNA that way. And we make a replica of the RNA, now in DNA form. So then you can use DNA polymerase to make the second strand. And what we end up with is double-stranded cDNA, double-stranded complementary DNA. Now, using basically the same tools, we clone this into a vector, and we now have produced what's known as a cDNA library. And we're going to carry out a screen in the exact same way. We're going to make P32 probe. We're going to plate out the cDNA library. We're going to do colony lifts, wash them, and start screening. The real trick here, though, is You might have a genomic chunk from an organism and you want to get the messenger RNA or the gene cDNA from another organism, or even the same organism. You got a genomic clone that you're interested in, and you want to find the gene associated with this that's being transcribed, the transcribed gene. And here's the rub. With genomic cloning, you know statistically, at least you go into the experiment knowing statistically, that if you do 3x coverage, you really are pretty likely to find your genomic clone. If you've got a gene that is expressed at very low levels or in a very narrow time frame, you may have to screen a bunch of cDNA clones to find your cDNA. So while we can give you a pretty good statistical analysis of how many clones to screen to get a genomic clone, all we can do with a cDNA library is tell you to keep at it and don't come back until you have the darn clone. I'll see you 2 or 3 months from now, next year, just keep screening. And so people have spent the better part of their lives screening for cDNAs. You can end up making cDNA libraries that don't have the gene at all in them. You can have cDNA libraries that aren't very representative. It's a bit of an art. You can do this now with PCR by using reverse transcriptase to make a cDNA bank that you're never going to clone out. You're just now going to use that as a target for PCR, and that works a little bit better, but you're limited, you're constrained by primary sequences and things you know. Whereas here, you might have a piece of DNA that is from a sequence that's from the very conserved part, and you can pull out a large piece that you're sequencing it and get some information. So now, the cDNA provides you with the actual piece of gene that is transcribed and processed and ready to be translated. And so now you can take the cDNA and put it into bacteria, because of course bacteria can't carry out exon intron splicing. They can't process messenger RNA. But you can now have bacteria take that eukaryotic gene, put it under the appropriate bacterial promoters, and bada bing, you can create fermenters that are producing your gene. So insulin is being produced this way. We have the cDNA clone for human insulin, taken from the RNA for insulin, and placed into a bacterial expression system, and you're off to the races. Now, with these two things in hand, we're now able to carry out some really good genetics experiments by looking at the splicing and the splice isoforms of a gene. We can look at the control sequences. This is the critical parts to any kind of good molecular genetic experiment and carrying out gene identification is having a genomic clone and having a cDNA clone. You got that, and you're set up pretty well. So now let's kind of step back and think about how we can compare this technology and these ideas to classical genetics. So if we have a show of hands, how many of you know about Mendel's laws of inheritance? And we got a couple of things that we can talk about. One idea is, one of the Mendelian laws is the notion of what's called independent assortment. And honestly, this is where molecular genetics kind of like zooms by Mendelian genetics. The law of independent assortment comes about from the observation that some genes are, quote, linked, and some genes are not linked. What we know is, if you've got two genes that are on the same chromosome, you get one, you get the other. It's like if I give you a stick, and I tell you, okay, the stick has two ends, here's the stick, do you have both ends? Yep. Wow, that was exciting. But that is one of the things that was observed in the early work, and we can establish this notion that is called linkage. So some genes are going to assort independently, meaning they're on different chromosomes. Other genes show a linkage association. And this comes, you had to have some way of labeling these different genes. It could be an eye color, it could be some kind of phenotype, whatever. And now we can do it by sequence, but regardless, we've got linkage. And so genes that are inherited always together are said to be closely linked or tightly linked. And genes that are inherited together better than 50%, but not all the time, are not very tightly linked. And what can start to throw this thing off? Linkage, we can diagram it like this. We've got a chromosome that has gene A and gene B. And then we've got another chromosome that is carrying gene C. All right. What we can observe is, if you've got markers for genes A and genes B, is they are often assorting with each other. If you get A, you're going to get B. And then the mutation in C might assort differently, because just remember, this is a haploid representation. We've got the diploid, and so here is we'll call it C prime, and the same thing with this chromosome. We got A prime and B prime, and we can assign different mutations and things to these. But the gametes can assort in whichever way they want. You could have this chromosome with either one of these, this chromosome with either one of these. Now, we say that two genes are linked if they tend to be inherited statistically better than 50%, because it's not always going to be 100% because of the following. We can end up, and it doesn't always occur with all organisms, but with recombination, in which we can have a break in the chromosome such that we end up producing what's called a chiasma. And once this is resolved, we actually have had a chromosome end exchange. All right, so what people began to do, and flies are really easy to do this with, is you start looking at different mutations, and you look to see if they're linked or if they're not linked. And from there, we can begin to look at recombination. And so, hopefully you remember from genetics or on the street, we can look at recombination frequency. And the general notion is the greater the recombination frequency, the further apart they are, the smaller the recombination frequency, the closer they are. Two very tightly linked genes typically will not have a recombination event occurring between them. Two genes that are spread apart from one end of the chromosome to the other are going to have recombination events occurring to them all the time. And you can even get double recombination events and things like this. So this creates this notion of linkage and what we call a linkage group. And these are genes that are very closely related, or very closely associated on the chromosome. It's a way of generating a map of the chromosome. And you can do an ordered linkage map. Now, one of the things that is interesting about this, though, is this began to establish the relative order of genes, and you can begin to place genes into different chromosomes based on their linkage and things and their inheritance. But there are times where you start looking at a linkage map, and then in the modern era you compare that to the sequence map, and it turns out that sometimes the linkage map is quite skewed because there are hot spots for recombination and there are other spots in the chromosome that just simply don't recombine. And because you're looking at recombination frequency, a hot spot can make recombination occur a lot faster there, a lot more frequently there, and it makes those two genes look like they're way further apart. And so it's not a great map of the genome, but it is a way. So this is the primitive, basic way we begin to map out a genome, is by developing linkage groups and establishing where genes are on a chromosome. So just as an example, in flies, you do a mutagenesis screen and you identify a mutation. And we'll talk about how you actually carry this, how you would do this in detail in a bit. But one of the first things you do is you map it to a chromosome by doing a linkage analysis. You look to see if it's inherited with specific markers or not. And that's a rather crude kind of Mendelian genetic trait way of mapping, but it works. So most genes encode proteins. And because of this, we can create mutations that are going to affect potentially the amino acid sequence of the protein and thereby affect its function. So the last little bit we're going to talk about is a genetic trick that we use. It's actually really important for thinking about genome complexity and all kinds of things. But it's this idea of what's called a complementation group. So let's just say we're carrying out a mutagenesis screen. And we're going to do this in flies, so we're going to be looking for lethal mutations. And you're going to say, we'll screen, a few thousand chromosomes to identify homozygous lethal mutations. Here's what we can find out and come up with. We've got, just as an example, these two genes, A and B, whatever. And we're going to create a mutation in A. Now, when we've got this in a genetic configuration in which we've got one mutated chromosome and the other chromosome pair is carrying a wild-type gene. For most mutations, the wild-type gene is going to cover for the other one. It's basically going to maybe has to upregulate to produce a little bit more protein, but regardless, it's supplying the function that's missing from this gene. And the easiest way to think about this, let's just think about having like what we call a null mutation, where we have a stop codon very early in the open reading frame. And you produce an RNA that's just non-functional. Well, the other good copy is able to take on. Now, it produces lethality, and we want to look at another gene, say, let's make it on the same chromosome. So we've got A and B, and we're going to make a mutation in B. And it's the same kind of recessive trait. So we carry it in a heterozygous state. Now, the question we want to ask is, are these mutations in the same gene? Now, given my excellent diagram, you can say, we already know that this guy is carrying mutation in A and this guy is carrying the mutation in B. Yeah, but you don't know that. We're going to carry out what's called a complementation test, and it works like this. You take these flies and you cross them. And what we look for is the flies that would be carrying this genomic organization, where we got the mutation in A, and then there's B. We got the mutation in B, and then there's A. Okay, so now what you got here is two different genes, two different mutations. This is going to be, under our standard logic, this is going to be a viable organism. And what we've established then is we've got a mutation in gene A and a mutation in gene B. And these two mutations, in this case, they complement each other. And this is known as complementation. If you listen very carefully, you're a very nice gene. Oh, thank you. You're a lovely gene too. They're very complementary. All right. Now, let's assume that we're doing a big mutagenesis screen and we got a lot of lethals, and some of them are going to be in the same gene, the lethal mutation. And so if we've got a setup like this, where we've got mutation A, and we'll call this mutation 531, and then here's B. And you've been dutifully screening your flies, and you got mutation A, you have mutation 1035, and B. You want to know, are these two mutations in the same gene? You carry out a complementation test again. And in this configuration, these two mutations are in the same gene, and we say that they will fail to complement. And if they fail to complement, that means that, at least from our logic standpoint, these two mutations, independent mutations, are in the same gene. What we can then begin doing is establish what we call complementation groups. So in this 1035 mutations that you've generated, you got, lots of lethals in there. You begin doing complementation groups with each one, and you establish the complementation groups. And so, in a sense, and more or less it works this way, a complementation group is a group of mutations that actually fail to complement, meaning that it's mutations in the same gene, independently developed mutations in the same gene. And one of the ways you can begin to ask the question, how many lethal genes are there in my organism, is to ask, how many complementation groups can you get? And I've been talking your ear off for almost 50 minutes. Thank you for your participation, your rapt attention. Go fix yourself a sandwich. I'm at least as good as Netflix. Yeah, whatever. Fine. One day this pandemic will end and we can do this in person, but not today. As I say, I'm working on a virtual lab thing for you. I found one online that's kind of dumb, where you point and click to stuff. I'm going to kind of build a little thing from that. But, complementation. We're going to talk a little bit more about different kinds of mutations on Thursday, and start getting into some genome organization type ideas, and see where we go from there. Thank you very much. Stay safe. I'm out of here.